Well, good afternoon. I'm Susan Shurin. I'm the Deputy Director of the NHLBI, and I'm delighted to welcome Dr. Kathy High as our Walls Lecturer today. Dr. High is the Director of the Center for Cellular and Molecular Therapeutics at the Children's Hospital of Philadelphia, where she's also an attending hematologist and the William H. Bennett Professor of Pediatrics at the University of Pennsylvania School of Medicine, and she's a Howard Hughes Institute Fellow. Last month, Kathy and Federico Mingozzi published a review article in the journal Blood entitled Immune Responses to AAV Vectors, Overcoming Barriers to Successful Gene Therapy. This is an elegant survey of the state of the art of gene therapy, particularly that using AAV vectors. Gene therapy products for the treatment of genetic diseases are now currently in clinical trials, and an AAV product has actually been licensed in Europe. The AAV vectors have achieved positive results in a number of clinical and preclinical settings, including hematologic diseases such as the hemophilias, in Gaucher's disease, hemochromatosis, and the porphyrias. So this is an extremely hot topic, and we're delighted to have Kathy here. AAV vectors are administered directly to the patient and therefore elicit a host immune response. And this is actually a major topic of Dr. High's current research, trying to achieve a comprehensive understanding of the determinants of the immunogenicity of these AAV vectors and the potential associated toxicities. In collaboration with global leaders in the field, she's engaged in immunosurveillance of ongoing clinical studies to provide the basis for understanding of the intricacies of the immune response in AAV-mediated gene transfer, facilitating safe and effective therapies for genetic diseases. Kathy has an interesting career trajectory. She majored in chemistry as an undergraduate and spent a year working in the biology of atherosclerosis in college. This led her to pursue an MD instead of a PhD in chemistry as she had originally planned. But we almost lost her to medicine because the emphasis on memorizing facts as a first-year medical student discouraged her. She took a year off to do chemistry research, during which her lab mentor in chemistry encouraged her to finish medical school. And fortunately for us, when she returned to medical school, things clicked and she decided to stay in medicine, but her solid grounding in science and in chemistry has served her well. Kathy's work regularly joins the boundaries between the bench and the bedside. She was drawn to hematology because of the extremely sophisticated understanding of the biology of many hematologic diseases on a molecular level. This matches her interest in biochemistry, chemistry, and molecules. As she was completing her hematology fellowship at Yale, scientists cloned the genes for factor VIII and factor IX. This feat presented her with an opportunity to sort out the molecular basis of disease, utilizing her chemistry training and her tremendous interest in the biochemical origins of disease. Her initial work focused on defining mutations of the clotting factor genes that result in hemophilia. This leads to ability to accurately identify disease carriers and permit prenatal counseling to women who carry the mutation, and it allowed her to analyze the relationship of the structure of the clotting factor molecules and their function. Kathy and her colleagues have developed models of hemophilia in mice and in dogs and work with dogs in which hemophilia occurs naturally. They've demonstrated that they can successfully treat the disease in mice and dogs by inserting a normal clotting factor gene into the animals, and she's now focused on adapting these therapies for humans. Her attempts at gene therapy have focused primarily on hemophilia B, which is the less common form of the disease. It affects about 3,000 men and boys in the United States. She's demonstrated in dogs with hemophilia B the principle that gene therapy can be effective in treating the disorder. Dogs that receive a single injection of the gene for factor VIII produce the clotting factor for more than 5 years. Human trials have proven to be a little bit more challenging. In small clinical trials using an AAV vector, a few patients produced therapeutic levels of clotting protein, but only for a few weeks. She's now exploring ways to modulate the patient's immune response just long enough for the virus to deliver the gene safely to the cells. So her major areas of investigation include the structure-function analyses of factors 7, 9, and 10, recombinant mutations, and the regulation of expression of genes. Please welcome Dr. Kathy High, today's lecturer, who will talk to us about genetic therapies for genetic disease, results and lessons from recent successes. I'd also like to remind you that we have a reception, which is sponsored by the FAES, in the library right after the lecture, and to invite all of you to attend. So, Kathy.
Well, I'd certainly like to thank Dr. Shuren for that wonderful introduction. I have to say that my career sounded much more interesting as she told it than it seems to have been during the living of it, at least to a certain extent. I thought that what I would do today, because this is an NIH-wide lecture, is talk to you not only about some of the hurdles that we've encountered in attempting to work out the details of gene therapy for hemophilia, but also talk a little bit about the work that we've done in collaboration with the ophthalmology group at the University of Pennsylvania of conducting a trial for a rare inherited form of blindness, Leber's congenital amaurosis. And then if there's a little time at the end, I'd like to say a little bit about the work that we've been doing most recently using genome editing to approach treatment of genetic disease. So I'll just remind you at the outset that, I work at a children's hospital, and that genetic diseases account for anywhere from 30 to 50% of all admissions to children's hospitals, and even for a very substantial proportion of admissions to adult hospitals. And yet, for many of these disorders, our therapeutic options are fairly limited, and that one of the hopes of the Human Genome Project was that it would provide a platform from which we could develop gene therapies to expand therapeutic options for people born with genetic disease. The goal of all gene therapy studies is essentially twofold in the setting of genetic disease. One is to achieve long-term expression of the donated gene, and the other is to express it at levels high enough to have a therapeutic effect. And all gene therapy strategies can be described essentially in terms of three components. First, the gene to be transferred in, the transgene, which in the case of genetic disease is not usually under dispute, in contrast to the situation in many acquired disorders. Of course, in addition to supplying the missing gene, one can do variations of this strategy where you supply instead a downstream gene product, or something that enables an alternate pathway, for example, 7A for somebody with hemophilia. But in general, this is spelled out clearly in genetic disease. We needed a vector to deliver the donated DNA, and then some physiologically appropriate target tissue into which it is introduced. So, for example, hematopoietic cells for sickle cell anemia. And to achieve long-term expression, one can choose from essentially two different strategies. One is to use an integrating vector and to introduce it into a stem cell, and from there it is passed on to every daughter cell, and one can achieve long-term expression on that basis. The other strategy, which is the one that we've been pursuing, is to transduce long-lived post-mitotic cell types. These are cell types in which, as long as the donated DNA is stabilized, it does not necessarily need to be integrated. One can achieve long-term expression simply because the cell itself is long-lived and is not going to divide. So, as you may guess from the picture here, many of these strategies using integrating vectors have used as their target hematopoietic stem cells, which lend themselves well to being extracted from the patient, manipulated in the laboratory, and then returned to the patient. Whereas most of these long-lived post-mitotic cell types, including, for example, cardiac muscle, skeletal muscle, cells in the brain and the eye, do not lend themselves well to being extracted from the patient and returned, but rather require that the vector be delivered in vivo. And so that, of course, predicts one of the major problems, which is that if you are going to introduce a viral vector directly into the patient, you are going to be grappling at close quarters with the human immune response. So all of our work has been with AAV vectors, and these have been engineered from a member of the parvovirus family. These are non-enveloped viruses with a linear single-stranded DNA genome of about 4.7 kilobases. And there are a number of features that make it attractive as a gene delivery vehicle, and I think it would be fair to say that of all viral vectors, it's one of the simplest, which is generally a positive feature. But as a vector engineered for gene delivery, it is non-pathogenic because wild-type AAV is non-pathogenic. It is naturally replication defective, again, because wild-type AAV is replication defective. And the DNA introduced by AAV vectors is stabilized predominantly in a non-integrating form. So problems related to insertional mutagenesis are minimized. And in the mid-1990s, two different groups described the result that a recombinant AAV vector introduced into skeletal muscle in mice could direct long-term expression of the donated gene in an immunocompetent animal. And since then, it has become clear that AAV vectors in their multiple different naturally occurring serotypes and in genetically engineered forms as well have tropism for over a wide variety of cell types, including a number of post-mitotic cell types that are long-lived, as you see in this list. So to take a little bit closer look at the vector itself, the wild-type virus encodes two types of genes, genes required for replication and genes that encode the capsid. The virion, the recombinant virion, is composed of essentially a wild-type capsid, which has 48 molecules of viral protein 3 and 6 each of VP1 and 2, all encoded in this capsid sequence. And then instead of the viral genome, that's replaced with the therapeutic gene of interest under the control of an appropriate promoter, so that the final recombinant virion with a diameter of about 25 nanometers is composed about 74% of protein, which is the viral capsid, and 26% of DNA. And so what you essentially have in this recombinant virion is a highly ordered set of proteins, the capsid, that enclose the DNA, which is the therapeutic agent. This material is dosed in vector genomes per kilogram of body weight of the recipient, and since it's pretty difficult to fight physics, you can't package more than about 5 kilobases of DNA into the AAV capsid. And there are a lot of different ways of producing AAV. This highlights the method that we use at Children's Hospital, and essentially 3 different plasmids, one encoding the gene of interest cloned between the viral inverted terminal repeats, another encoding the rep and cap genes, and the third providing the adenoviral helper genes needed for replication, are introduced into 293 cells, and subsequently the recombinant virions can be harvested either from the supernatant or from the cell lysate, and one can either use all of the product purified from the cell lysate or separate it into the fulls and the empties, which still constitute a substantial proportion of what's generated in most production methods. So we usually take the empties out, and I'll say more about that in a bit. And as Dr. Shuren mentioned, I want to be sure to say that the first gene therapy product in an ICH country, you see me later if you don't know what that means, but anyway, it means basically the United States, Europe, or Japan. The first AAV product was licensed last year in Europe. This is a product for lipoprotein lipase deficiency, and I think it's important to point out that this product is based very heavily on work done here at the NIH by Rob Cotton and his colleagues in NHLBI. So it does mean that AAV products are beginning to appear on the scene, so that's good for all of us. So As Dr. Shuren mentioned, I've had a longstanding interest in hemophilia. I'm a hematologist, and I've been working for a number of years to try to develop an AAV gene therapy for hemophilia. And since clotting factors are normally made in the liver, liver has been an important target for us. Through the years, a number of obstacles have had to be overcome to make AAV gene therapy in the liver a reality. One of the first was that in the early days, we could make enough vector to do mice, but not to do hemophilic dogs or people with hemophilia. So that was one of the first hurdles. In the initial trials in which AAV was introduced into liver, we encountered a problem that had not been foretold by animal models, and that was that the vector was detected in the semen of the first subjects, and we had to work out strategies to mitigate the risk, to track that, and to reduce the risk of a piece of vector DNA being incorporated into an offspring. I'm going to spend a little time talking to you about the issues that have arisen as a result of the human immune response to AAV. And finally, of course, for any new class of therapeutics, one issue that must be addressed is what is the long-term safety related to the use of the product. And for AAV, the primary long-term issue is likely to be related to integration. I told you that AAV is a predominantly non-integrating vector, but anytime you introduce foreign DNA into a cell, there is a possibility that some of it will integrate into the host cell genome. This is true for AAV as well, and the question is, will this have long-term effects? So I'm going to spend a little bit of time talking about the problems related to the immune response, because I think these have been the primary short-term problems that have had to be solved to make gene therapy for hemophilia a reality. And to state these quite simply, from a genetic engineering standpoint, AAV vectors are essentially a highly ordered set of proteins that access receptors on the cell surface and deliver DNA to the nucleus. The DNA persists, the delivery proteins are degraded, and you get long-term expression. And that was observed over and over again in many large animal models before any human trials were conducted. However, AAV vectors, viewed from an immunologic standpoint, have been engineered from a virus that the immune system has previously encountered. And so it's been important for us to understand exactly what are the human immune responses to the recombinant virion so that we can manage those responses and get a therapeutic outcome. So I would say that our lab over the last several years has been focused on the following questions. From a biological standpoint, what host immune response is encountered by a recombinant virion that fails to synthesize any viral antigens, but also fails to synthesize the viral genes that normally downregulate a host immune response? And from a medical and therapeutic standpoint, can the host immune response that's generated by a recombinant virion be controlled so that gene transfer occurs and the host is unharmed? And I will say that this latter question is made more complex by the fact that for every target tissue, the human immune response is tissue specific. And so the answer to these questions is essentially a tissue specific answer. So you have to work it out one tissue at a time. So let me just say a word or two about hemophilia. It's the X-linked bleeding disorder that arises because of mutations either in the gene for factor IX, which is an enzyme, or for factor VIII, which is the coenzyme for the same step in coagulation, and that clinically these are indistinguishable and are characterized by frequent bleeds into the joints and soft tissues, but patients, especially those who are severely affected, can also have bleeds into other closed spaces, such as the intracranial space, where the bleeds can be rapidly fatal. Hemophilia A affects about 1 in 5,000 male births, and hemophilia B about 1 in 30,000 male births. The other point that's important for gene therapy is this: the largest category of patients with both diseases are severely affected, with less than 1% factor VIII or factor IX. But we know from the natural history of the disease that if the circulating levels are even slightly better, the patient is spared most of the life-threatening bleeds and many of the joint bleeds as well. And if a person's level is raised to around 5%, this person may go through childhood without ever being diagnosed, so it's a relatively mild phenotype. So we know that for what we're attempting to do, that if we can elevate the levels by just a few percent, we're likely to improve the phenotype of the disease. And right now, of course, hemophilia is treated by intravenous infusion of clotting factor concentrates, either plasma-derived or recombinant. Typically, children are treated in a clinic until they reach the age of about 4. You get these infusions 2 or 3 times a week. And starting at around 4, usually the parents can be taught to do the intravenous infusion, and subsequently children learn to do it themselves. These products cost around $200,000 a year for an adult male who is infusing himself 2 to 3 times a week. And because of these costs, it's estimated that they're available for about 20% of the world's hemophilia population. And if you look at the rest of the world's hemophilia population based on per capita GNP, this graph displays children with hemophilia compared to adults with hemophilia as a function of the GNP. And you can see that in the Western world, there are many more adults than children, but that as the GNP falls, it really becomes progressively a lethal disorder that kills most people before they reach adulthood in resource-poor nations. Okay, so I'll skip over a lot of preclinical work that we and other people had done in mice and dogs, and I'll just say that by the late 1990s, we had the following result in the naturally occurring hemophilia B dog model, that a single injection of an AAV vector introduced either into skeletal muscle or into the liver in a dog would result in long-term expression. We got better levels by going into liver, I think because liver is better at secreting a gene product directly into the circulation. But you can see that these animals that were dosed at time 0 were still expressing therapeutic levels 140 weeks later. We followed some of these animals for as long as a decade, and it's a very long-lasting effect from a single infusion of vector into the portal vein or the hepatic artery. And similarly, when we did studies in humans where we introduced an AAV2 vector into skeletal muscle in men with severe hemophilia, we recently had the opportunity to go back and sample muscle in a patient who had been injected with AAV2 factor IX 10 years earlier, and there was still factor IX being expressed in the injected muscle. This is immunohistochemical staining for factor IX. However, in the first subject in a liver trial to receive a therapeutic dose, instead of seeing long-term expression as we had in hemophilic dog liver and in human skeletal muscle, instead we saw, as you see diagrammed here in red, expression at a therapeutic level in this patient for about 4 weeks, and then he slowly lost factor IX expression until he returned to his baseline of less than 1%. This was a time course that we had never seen in a series of different animal models, and it was accompanied by another finding, which was a transient transaminase elevation that occurred in tandem with the loss of factor IX expression. So the time course of this suggested an immune-mediated response, and we were actually subsequently able to develop reagents to look at this specifically. This is using a pentamer to examine the population of capsid-specific CD8 T cells in a subsequent patient who was infused with AAV factor IX. And you can see that what happened in this patient, he didn't get detectable factor IX expression at all, but he did experience this transaminase elevation. And when we use this pentamer to look at the population of capsid-specific CD8 T cells, we could see that that population of cells expands after vector injection. and then it contracts in a time course that matches the rise and fall in liver enzymes. And in other experiments, we were able to show that the specific epitope that was recognized by the patient's CD8 T cells was highly conserved in a number of different AAV capsids, and that they evoked the same response when the patient's lymphocytes were exposed to the appropriate epitope. So there was cross-reactivity with a number of different AAV serotypes for this particular epitope. So our working hypothesis is shown here, that the capsid is essentially a preformed antigen that after the vector enters the cell in the endosome and then escapes from the endosome, it eventually uncoats and the DNA goes to the nucleus and begins making factor IX. But some of the capsid is left behind in the cytosol, and we know based on work that was done by John Engelhardt and others that that capsid left behind undergoes proteasomal processing and can eventually be transported into the ER, where it gets loaded onto class 1, displayed on the surface of the transduced hepatocyte, and then makes that cell a target for circulating CD8 T cells, which, because humans have memory to AAV, whereas other species do not, results in the destruction of the transduced cell. And based on a variety of other experiments, we have been able to demonstrate that the level of antigen presentation is dose dependent. So the higher the dose that is delivered into the cell, the greater the likelihood that this transduced hepatocyte will be detected and destroyed. So a number of other hypotheses were proposed to explain that series of findings. And one was that the vector had, during manufacture, packaged the RepCap plasmid so that the patient was continuously expressing capsid. One would predict then that short-term immunosuppression would not work. There was another hypothesis that there were alternate open reading frames in our expression cassette, and that the immune response was actually directed toward those. And there were predictions that if we switched serotypes, we would avoid the response because the AAV2 serotype transduced antigen-presenting cells more efficiently than some other serotypes. Our hypothesis about preformed capsid led to the prediction that short-term immunosuppression would work as an approach if one could simply block the immune response until the preformed capsid was degraded in the cell. One could perhaps see long-term expression. We did a series of experiments to try to support that hypothesis, and I'll just point out maybe a couple of those. We eventually cloned the T cell receptor from a capsid-specific CD8 T cell, and then you're essentially able to use this as a detecting reagent to detect peptide MHC complexes that are carrying peptides derived from the AAV capsid. So this is some of the data that I told you that we had that suggested that this immune response was dose-dependent. If you take that T cell receptor and clone it into a T cell line that expresses luciferase in proportion to the degree that it recognizes its cognate peptide MHC complex, then if you take human hepatocytes and transduce them with AAV capsid, then you can show that at increasing levels of capsid multiplicity of infection of the vector, you get progressively increasing expression of luciferase. So it demonstrates that the amount of antigen displayed on the surface of a transduced cell is dose dependent. Okay, so the other important thing that we learned in that initial trial, and the work that I told you about was done, over years after that first trial, but the other important thing that we learned in that first trial, these two patients both got the same high dose of that first vector, and one of them had detectable expression for a period of weeks until the CD8 T cell response supervened, whereas the other one got very little detectable expression and by 4 weeks was down to 0. This person had pre-treatment neutralizing antibodies to AAV, fairly high titer, 1 to 17, whereas this person had a low titer of neutralizing antibodies. So the other important thing we learned then was that this pre-existing humoral immunity to AAV would be blocking if we chose to infuse the vector through the circulation. And we had considerable data in the original muscle trial that said that if you're going through skeletal muscle, these antibodies aren't a problem, but clearly if you're going through the circulation, they would have ample opportunity to neutralize the infused vector. This is from a study done by Jim Wilson. These are percent of the population with neutralizing antibody titers of greater than 1 to 20. And you can see that the percentage in the population is fairly substantial across 4 continents. And that's 1 to 20. That's fully blocking. So really, we're interested in the population with even lower neutralizing antibody titers. So the second AAV factor IX trial was conducted at University College London and St. Jude Children's Research Hospital. And so the solution they proposed to those 2 problems with the immune response that had been seen in that first trial were shown here. First, just exclude everybody with neutralizing antibodies to AAV. So that's not a good long-term solution because it involves too many adults, and there are a lot of adults with hemophilia, but it's a good short-term solution. And then they added a provision that a short course of steroids would be administered if the liver enzymes rose or the factor IX level began to decline. So in addition to that, they did a number of things to make their vector more efficient. They used a serotype, AAV8, that has a strong tropism for liver. They used a self-complementary vector design, so normally these vectors are single-stranded. Theirs was essentially already double-stranded, so it didn't have to rely on the host cell machinery for second-strand synthesis, and it was codon optimized. So the goal and the expectation was that it would be possible to infuse a lower dose and still get therapeutic levels of expression, and that perhaps at this lower dose, the T cell response would not be encountered at all. And in fact, in the first subject that was infused in that study, this appeared to be the case. This patient was infused now over 3 years ago, and he, after infusion of 2 times 10 to the 11th vector genomes per kilogram, his circulating factor IX level went from less than 1% up to 2%. That's a low level. It's modest, but it's definitely different from less than 1%. And this patient has been able to reduce very substantially the amount of clotting factor he uses to manage his disease. So for him, 2% really mostly removed the need for prophylactic factor IX infusions. However, as dose escalation continued, what was seen at this dose, 2 times 10 to the 12th, initially the patient had a circulating level of 6 to 8%, which is great. That really converts his severe hemophilia to mild. But starting at about 7 weeks after vector infusion, the liver function test, which had been completely normal, began to rise, and then they rose very quickly over the period of a few days. And that was, of course, the predetermined trigger for initiating an 8-week course of prednisolone. And that rapidly renormalized the liver function tests and stabilized the factor IX level, and it eventually stabilized at between 1 and 2%. So we had the opportunity to study the lymphocytes from this individual using interferon gamma ELISpot, and what you can see is that the time course tracks very closely with what happened to the liver function test. So you begin to see some modest level of response above the baseline at about 5 to 6 weeks, and then at 8 weeks it shoots up to a markedly positive response in parallel with this rise in the liver enzymes. And then, of course, after the initiation of prednisolone, these cells rapidly disappear from the circulation. So the clinical investigator in London, Ted Tuddenham, learned a great deal from that. And if you look here at the frequency with which he checked the liver function tests after that previous patient, you can see that he was extremely vigilant. And as soon as these started to go up at all, he started the patient on prednisolone, and that patient has stabilized now 2 years later with a circulating level of around 5%. Again, what we saw here was a very modest response initially, and then it shoots up at 9 weeks, just at the point where it appears that the LFTs are starting to rise even very modestly. So since then, they've treated several more patients, and this was presented at ASH, and you can see that most of these people are stabilized in the range of 4 to 6% now, and so that's a very exciting result. 4 of the 6 people needed that course of steroids. 2 of them went through with never raising the LFTs at all. So one of the important goals for the field is to try to determine what percentage of adults are actually going to need this course of steroids, how many people will actually have this response. And of course, another way to look at it is, well, the easy way to do it is just if you know exactly when this is going to happen, can you just give the 4 weeks of steroids and not worry about tracking it? So I think that what's important about that high dose cohort, it's been possible to identify a dose that leads to long-term expression at therapeutic levels. The transaminase elevation is asymptomatic, but it serves as a marker of the cellular immune response that then triggers the investigator to start the course of steroids, which seems to rescue or stabilize the transduced hepatocytes, even though administered for a relatively short time of 4 to 8 weeks. And I think that for most hemophilia patients, the risk, at least short term, of this series of events would be far outweighed by the benefit of long-term expression of a level of factor IX in the range of a few percent. So what I would say about where the field is now in developing AAV-mediated gene transfer to liver, we understand, I think, much better the problems posed by the humoral immune response to AAV and by the cellular immune response, and we believe that at least at the doses shown here, 2 × 10⁻¹² vector genomes per kilogram, it's possible to control the cellular immune response with a short course of steroids. And so the question about the long-term safety of the product, I think, will only be answered by continuing to follow both large animals and human subjects who have been infused with vector. And so obviously all of these patients are followed for life. So let me just say a word about what we've been trying to look at as a strategy for the 40% of the adult population with pre-existing antibodies to AAV. So we got our idea from this by looking at comparing the results of the first 2 trials, the AAV2 factor IX trial, which had a single-stranded DNA vector, and the AAV8 trial, which had a self-complementary. But in the end, the doses in these 2 trials ended up being about the same. And at the high-dose cohort, at least before the T-cell response supervened, you can see that both of them led to a similar circulating level of factor IX, which would suggest that the potency of the 2 vectors was similar. However, in the AAV2 trial, there was no expression at the low-dose cohorts, whereas in the AAV8 trial, there was expression, at least a modest level, at the low and medium dose cohorts. So, what was the difference? What accounted for this? Well, was it the serotype? Was it the difference in the DNA conformation? We wondered about another difference between these 2 preps. The prep that was administered in the UCL St. Jude trial actually had a ratio of about 10:1 empties to fulls, whereas the production process that was used to make that vector, which was actually made at Avogen, separated the fulls from the empties. And so what was infused into these 2 subjects differed in that way. And so we wondered whether the empty capsids might in fact serve as decoys to absorb circulating antibodies to AAV resulting in better transduction efficiency, particularly at the lower doses. So we carried out a series of experiments. These were done by Federico Mengozzi and Xavi Anguilla, and they used a mouse model. The mouse was injected with human IVIG at a range of doses to mimic neutralizing antibody titers that are encountered in the human population. And then the mouse was injected with AAV factor IX vector with no empty capsid or with a 10-fold excess of empties. So here's what you see. If the mouse has no neutralizing antibody titer, it doesn't matter whether you add empties at 10-fold excess or you don't, you get the same level of factor IX expression. If, however, you give the mouse a low titer neutralizing antibody to AAV, then if you don't add any empties, you really don't get much expression. And if you add a 10-fold excess of empties, you get very nice levels of expression. And so Federico went on to do a series of experiments where he just gave mice progressively higher neutralizing antibody titers of using IVIG, and then showed that by adding progressively higher levels of empties to the final formulation, that you could restore levels up to that seen in a naive mouse, but the higher the antibody titer, the higher excess of empties is required to achieve that effect. And if the mouse has a very high neutralizing antibody to AAV, you cannot overcome it because you start to inhibit receptor-mediated uptake with the vast excess of empties. So, so the way to make this even safer, of course, is to alter the receptor binding site so that the empties, when they're infused, can circulate, but they can never gain access to the target cell and contribute to the burden of capsid that's going to be processed and presented on the surface of the transduced hepatocyte. So that's another strategy that we're interested in. And we think that eventually, for use of this kind of strategy in people with hemophilia, you're going to have to couple a sensitive assay for the neutralizing antibodies to a personalized final formulation of the product. Okay, and so so we are actually testing this in our currently ongoing trial at Children's Hospital. And so in that trial, we'll be using the same expression cassette that we had used in the first AAV2 trial, but in an AAV8 capsid. And then the final formulation will be linked to the patient's neutralizing antibody titer. So this trial is open and ongoing at Children's Hospital and the University of Pittsburgh now. So I think for the field as a whole around hemophilia, obviously there are a number of very important questions. What will be the duration of expression? In the UCL St. Jude trial, there is now over 3 years of expression in the first subjects with observation ongoing. Will it be possible to re-administer, and what will be the role of neutralizing antibodies, which always rise after vector infusion? Will it be possible to pursue the same approach for hemophilia A? And certainly for protein therapeutics, there's a much greater risk of immunogenicity in factor VIII deficiency compared to factor IX deficiency. And then, of course, what will be the long-term or late side effects of this type of therapy? We have followed over 77 dogs with severe hemophilia A and B for periods of over 10 years and have not seen long-term side effects related to this therapy. And then the subjects from the first trial were all infused between 2001 and 2004. They're part of a long-term follow-up study with no evidence of problems. And will it be possible to extend this to the pediatric population? Okay, so what I'm going to do, I actually see that, as usual, I have been overly ambitious about what I can cover. So what I would like to do, though, in the remaining minutes is turn to a situation. This is an application that we've been working with over the last 7 years with Jean Bennett and Albert McGuire in the Department of Ophthalmology at Penn. And of course, if you spend a lot of time worrying about immune responses to your vector and its transgene product, it's always tempting to think about a target tissue that is relatively immunoprivileged. And of course, that's what the subretinal space represents. And Jean had worked for a number of years on a particular genetic form of inherited blindness, Leber's congenital amaurosis, due to a mutation in the gene that encodes retinal pigment epithelium 65 kilodalton protein. What a mouthful. Okay. But it's an autosomal recessive defect, and all Leber's is characterized by the early onset of retinal degeneration, and these children are generally diagnosed in infancy or early childhood when the parents notice that the baby does not track visually. 10 to 20% of the cases of LCA are due to mutations in this RPE65 gene, and there's a naturally occurring dog model of this. And this is a list of genes that, if they contain mutations, can lead to LCA, and as I said, some modest proportion of all these cases are due to mutations in RPE65. So from an anatomic standpoint, the mutation affects the retinal pigment epithelial cells where RPE encodes an isomerase. And so normally when light strikes the retina, you get this cis-to-trans isomerization reaction that then starts an action potential that goes back through the optic nerve. But eventually this transretinal is transported back out of the photoreceptor into the RPE cells, where it must undergo this isomerization to generate cisretinal again. And if you do not have the activity of this enzyme, then the visual cycle is broken and blindness results. So Jean had shown in an animal model, this naturally occurring dog model, that subretinal injection of an AAV vector expressing RPE65 could restore vision in these animals as long as the animal was treated relatively early in life. And the reason for that is that as the RPE cells, that full function of the RPE cells is really required for the photoreceptors to survive, and that eventually these will degenerate, and then it's not possible to restore vision even if the enzyme is reintroduced. And she and her colleagues were able to flesh that out a little more clearly in a mouse model. So let me just say that among the many things that we did to get ready for this trial were device studies, and this is one of the devices approved for subretinal injection in the United States. And one of the points that we learned as we got ready to do a clinical trial for this is illustrated here, and that is that if you don't put a small amount of surfactant into your final formulation, that the low concentrations of vector used in this setting result in most of the material sticking to the delivery device and the delivery of substantially less than was initially loaded into the syringe. And so we do put a small amount of surfactant into our final product. So after completion of a series of studies to optimize the vector and to decide on the final formulation, this trial was initiated at Children's Hospital. And we initially limited it to children at least 8 years old, but young adults not more than 27 because of that dog data. And the dose escalation design included 3 subjects in each of 3 dose cohorts. But because children were going to be included in the trial, the IRB insisted that even the first dose that we use was one that showed efficacy in more than 90% of affected dogs. And the reason for that, of course, is that if children are involved in clinical trials, if it involves more than minimal risk, which certainly this sort of injection in the operating room does, there must be the prospect of direct benefit for that child. So we had to start at a dose that seemed to work in dogs. And so after extensive baseline testing, subjects are enrolled in the study, and the worst of the 2 eyes is injected. So the first subjects were enrolled in the fall of 2007. And this shows the data on the first 12 people who were in the phase 1/2 study. They've now been followed for periods of over, the longest followed have been followed for over 5 years, about 5.5 years now. So it's certainly been safe. And all of these people, we collected data on visual acuity, functional MRI on some of them, visual fields, pupillary light response, and performance on a mobility assay that was set up in the ophthalmology clinic that has a series of obstacles that the patient has to step over and around to get to a door at the end. And I'll just say that the results of the phase 1/2 study for the injection of the first eye showed that for 9 of 10 evaluable subjects, the injected eye develops a stronger pupillary light reflex, and no uninjected eyes develop a stronger pupillary light reflex. For 6 of 6 evaluable subjects, there was increased light sensitivity in the injected eye, and that didn't happen for any of the subjects in the uninjected eye. 4 of 8 evaluable subjects demonstrated improved navigational ability using the injected eye, but nobody did that for the uninjected eye. And similarly, 50% of the patients showed improvement in visual acuity, more than 3 lines on the eye chart in the injected eye, whereas nobody did that in the uninjected eye. So, you know, if you've never seen these videos of children going through the mobility assay, they're very impressive. I'll show just a minute or two. What you will see in this first video is 3 months after vector injection into the worse eye. The subject first has his injected eye patched, and he's trying to go through the mobility course using his uninjected eye, which was previously his better eye. And what you see, he's supposed to be following the black arrows on the floor, and even at the very beginning, he wanders off the course. And that's Gene Bennett in the background redirecting him. And I think, you know, you don't have to watch it for very long to understand that this is really blind behavior. And then when the patch is applied to the uninjected eye and the patient is asked to go through the mobility course using his injected eye, you can see a very marked difference in his performance in these two situations. So to move this forward, the FDA was interested in knowing the answer to whether it was safe to inject the other eye. And so we initiated that study in November of 2010. We had to write in the consent form for all of these people that it was possible that they would develop an immune response that would wipe out the gains in the first eye. And so fortunately for us, 3 of the adults were willing to sign that consent form and to undergo the injection. There was an 8-week stagger between subjects to try to monitor for any evidence of an immune response, which did not occur. And because it appeared safe in these first 3 subjects, it was possible to do the remainder of the individuals, except for one individual who had glaucoma in the contralateral eye, so he couldn't undergo injection of the second eye. Currently, we're engaged in a phase 3 study of this, and I think that you can understand that one of the controversial issues was what to use as the control. The FDA was concerned that if we used one eye injected and the other eye's control, this controls for any effects due to the specific mutation that the patient has. All of these people had different mutations in their RPE65 gene, but of course the product will not, in the end, be administered to one eye. It'll be administered bilaterally. So in the end, what we decided to use was that subjects, if they are deemed eligible, if they meet all the eligibility criteria, they're then randomized 2-to-1, either to be injected bilateral, non-simultaneous injection within 1 week, or they go into a control group, and the control group undergoes all the same testing, but 1 year later is permitted to cross over to the treatment group. This study is being conducted both at Children's Hospital and at the University of Iowa, and enrollment is proceeding very nicely, and we hope to have finished enrolling that study by early 2014. Okay, so I think what I'm going to do now is unfortunately, I'm not going to have time to talk about the work we've been doing with genome editing. This is a very interesting approach, but let me just summarize and say this, that in vivo gene transfer with AAV vectors is a clinical reality. In fact, it's a licensed clinical product in Europe. Recombinant AAV vectors are not viruses. I always want to try to get that point across. But these moieties encounter or evoke immune responses in humans. It's been important to try to understand those, to characterize them, so that they can be appropriately managed. But you know, for those of you who do allogeneic bone marrow transplantation, I have to say understanding and managing immune responses with AAV vectors is way easier than transplanting bone marrow. And each target tissue or route of administration represents a unique problem in terms of immune responses. But I think we now have a very nice platform for delivery to the subretinal space and to the liver at the doses that have been used so far, and that provides much scope for further development in the field. So I want to just say that in our group at the Children's Hospital, the work on immune responses has been led by Federico Mingozzi, who has recently taken a faculty position at Genethon, and also by Etienne Bosner-Chakarian, Danny Huey, and John Fenn, as well as lots of other people. Look, Sheng-Zhen is so important. I listed her name twice. Okay, so everyone knows she runs our research vector core. That our collaborator for all the studies in hemophilic dogs has been Tim Nichols at UNC Chapel Hill. That Jean Bennett, Al McGuire, Dan Chong, and others at Penn Ophthalmology have been our partners in the LCA2 trial. A number of other investigators at Children's Hospital involved either with clinical grade vector production or with preclinical and clinical studies in hemophilia, and Greg with LCA2, our collaborator on many of our immunology studies, Gundi Erdel. The AAV2 clinical trial, many collaborators at Stanford, Abidjan, University of Pittsburgh, Royal Prince Alfred Hospital in Sydney, and Campinas in Brazil, and at the AAV8 Factor IX trial conducted by Amit Nathwani, Ted Tuddenham, Art Nienhuis, Andy Davidoff, and Ulrika Rice at UCL and St. Jude. Okay, thank you very much for your attention, and I'm happy to take any questions if there's time. I don't know, do we take questions here?